MBSI Bioinformatics course. So first off, I'll talk you through the structure of our workshops. So in workshop 1, this workshop will be introducing you guys to Biopython, and we'll be manipulating RNA, DNA, and protein sequences. And basically, it's giving you guys the skills and tools you need to have a go at the next few workshops. In workshop 2, we'll be looking at gene alignment, so how we actually align different sequences and the algorithms we use to do so. And in workshop 3, we'll have a go at aligning multiple sequences and creating evolutionary phylogenetic trees here. All right, so in this workshop, I'll just give you guys a brief overview on the new topics and kind of discuss, why this is important and why we should do this. And then for the rest of the time, we can work on the exercises in the worksheet, and the tutors will be coming around and helping you guys with that. All right, so this has been mentioned in the launch event already, but so what exactly is bioinformatics? Well, it's a multidisciplinary field involving biology, genetics, biochemistry, computer science, mathematics, statistics, and more fields. Basically, it's the usage of computational and analytical tools to analyze and study biological and medical data, whatever that data may be. And the amount of data is exponentially growing, which means the need for bioinformatics is too. Some examples include modeling biological processes from experimental data. So, for example, if you have a reaction pathway, seeing the products of this reaction pathway and filling in the gaps. Inferring evolutionary relationships between species based on gene sequence samples. So shown here, you can see which species are more closely related to each other from the genes, and analyzing protein structures for clues in function and structural prediction from just the sequences themselves. So a bit of genomics in case you guys weren't familiar. So when I talk about sequences, what I mean is these letters here. So this is a DNA sequence. Sorry, this is a DNA molecule, and it looks like a ladder, and it has two halves to it. So the left half and the right half. So when I say sequence, I mean the order of which these bases or these letters here appear. So for example, if I read down this sequence, this would be A, G, T, C, A, T, G, A. So we only need to know one side of the DNA molecule because we know that A's bind to T's and C's bind to G's. So once we know one side, we'll know what the other looks like, or we know what the complementary side looks like. Okay, so why sequence genes? Why do you actually do it? So using COVID-19 as an example, we can identify new mutations and track variants. We can also track the global transmission routes, which is what this little graph here is. So starting from the bat COVID-19, it heads up to Asia and spreads throughout the world from there. You can develop rapid COVID tests, and you can also identify variants that may affect vaccine potency in the future. So some non-COVID examples include which genes are expressed under varying conditions, which strains of bacteria carry antibiotic-resistant genes, and also we're able to recreate facial features from just a drop of blood. So that's what this is. All right, so as for gene sequencing technologies itself, the cost and time of gene sequencing has greatly reduced. So costing up to 26 million to sequence one human genome in 2006, and also a few years to do so, to costing about 600 to sequence your own genome today for just a few hours to a few days. And yeah, the data used in this workshop will be in DNA sequences, although we'll transform the DNA sequences into RNA and proteins in the workshop themselves. So again, it contains 4 nucleotides, adenine, thymine, guanine, and cytosine, and this will be in long strings of these letters, basically. Some sequencing techniques include Sanger sequencing. So that's the older way of doing things, which is more accurate, although slower. And Illumina and nanopore sequencing, which can be considered as next generation sequencing. And that's the high throughput stuff that that costs less and we can do more of. All right, so to get from a piece of DNA that you've picked out of a cell to actually analyzing that specific DNA, we have to first sequence it and prepare it for sequencing. And there's a few steps in between. So first you have to prep it for sequencing, and that includes first fragmenting the DNA sequence, because when you pick out a piece of DNA from a chromosome, it could be hundreds of thousands, if not millions of base pairs long. And that's not really ideal for us. So we chop it up, then we repair the ends and apply special sequences called adapters to the ends of the fragments. And these are basically tags for the fragments themselves. And we can select the sizes of the fragments this way because each adapter is specific to that fragment that you want. And we amplify it, so we make more of the fragments that we actually want to have a look at. So then we sequence those fragments and we export them as a Fastq file, and we'll talk about that later on. And then from there, we have a look at the Fastq file and we apply quality control and analysis onto it, which is what we'll be doing in the workshops. And all right, so now I'll just go through the steps of Illumina sequencing, which is a pretty popular next generation sequencing technique. All right, so first off, you get your genomic DNA, which is DNA that you've picked out of a chromosome. And it's a long piece of DNA in 2 strands. First thing we do, we split the strand up into single strands. Then we use enzymes or special proteins to cut up the long DNA strand. Next step is we get adapters and we chuck them at the ends of the fragments. That's this step. So next we have this plate called a flow cell, and sticking out of that plate are little pieces of DNA that is complementary to the adaptive sequences that we put on. So the adaptive sequence at the bottom of the fragment binds to this adaptive sequence on the flow cell. So what you end up with are these fragments here sticking up from the flow cell. And we're only interested in one specific direction of the DNA strand. So for example, if this fragment here has a blue adapter on one end and a red adapter on the other end, we're only interested in strands where the red end is sticking up. So if there's any strands here with the blue end sticking up, we wash them away. And then we apply PCR and bridge amplification to get from single strands to clusters. And we'll see how we do that in the next slide. All right, so we'll read from the left side first. So you have your fragments, you attach adapters to those fragments, you attach them to the flow cell. And the flow cell also has fragments on the flow cell that are complementary to the adapters sticking up. So your fragment will bend over and attach the flow cell on both ends. And what we do is we synthesize the complementary strand of that fragment, and then we detach them from the flow cell, and then it attaches to another part of the flow cell, and then we synthesize that, and it attaches, and so end up with these clusters. Then we wash away the complementary strand, so we only get one direction sticking up. And then we apply what we call primers. So these special sequences here that attach to the adapter on the top of your fragment. So from there, we get special nitrogenous bases, special A, T, Cs, and Gs that fluoresce when you zap them with a laser. and we introduce them one by one. So for example, we introduce one base here. So you build upon this primer by one base, and then we zap it with a laser and see the colors that it fluoresces up. So for example, T's here fluoresce blue. So you get different clusters of blue, orange, green, and red on your flow cell. Then you introduce another base to it. So another rung of the ladder, zap it, you get different colors, and you keep doing that until you've gone all the way down the fragment. And yep, so here, so it starts at the top. So you introduce your bases and you go down and you zap each step with a laser, and you end up with this digital image here. And for each cluster, there'll be a majority color of that cluster, and that's how you can tell which base it's currently on. And yep, cycle repeated until all bases are counted. So a flow cell ends up looking something like this when you fluoresce them. So there is a ton of potential clusters you can have on a flow cell, so up to 1.6 billion clusters when you oversaturate it, which is not a good thing. So you want to have a bit less than that, so the machine can actually tell, the machine's not overloaded, basically. It can tell which cluster belongs to which. And yeah, this is why we call it high throughput sequencing, because we can sequence so many clusters just in one go. All right, so what kind of data are we actually getting out of this? So we're getting what's called a Fastq file or Fastq format. So this is just a text file, and it contains your multiple sequences of your different fragments. So here, we broke the DNA up into fragments that are 50 base pairs long, so 50 bases long. And yeah, so each sequence has 4 lines. The first line is the identifier tag, so which cluster or which sequence it belongs to. It has a quality score in base 33 or 64, so that's what this line is here. It looks like someone's smashed their keyboard too many times. And what that is basically a score from 1 to 64. Well, 1 in 33 for Sanger, 1 to 64 for Illumina. And that's basically telling us the quality of that particular base. So a score of 1 would be, you're better off just rolling a dice and getting the base of that. Whereas 64 is 100% certain that that is the base. And that's just based off of signal strength from your cluster reads when you have a look at the flow cell. And yeah, so your sequence here in A, T, C's, and G's, and your quality scores here are separated by a plus spacer, although sometimes they might use a different symbol. And that's just to separate the lines, make it look pretty. All right, so another format is FASTA. So it's similar to the Fastq file in that it can also contain multiple sequences, but these are more used for longer sequences, so that have been reconstructed from your fragments in the Fastq file. So these are longer sequences, and they are usually used to store reference data from curated databases. So not raw data, but data that's already processed. And it consists of one header, so one line, identifying the sequence, and then the sequence itself. So no columns, no blank lines, and the sequence can have numbers or spaces. So sometimes this sequence may be in amino acids for a protein, for example. And in that case, we use different letters and numbers. All right, so the third format that I want to talk about is Genbank. And that's a much more detailed, it's much more detailed than FASTA or Fastq in terms of the identifiers used. You see it's like a whole heap of descriptors for your sequence. And it's used almost exclusively for already curated and annotated data. So what I mean by annotated is just we know which parts of the sequence codes for something or like does something. And yeah, it usually almost always contains a single sequence. So I think now I'll actually look up the COVID gene and we'll have a look at its Genbank entry. So first off, if you want to have a look at if you want to get Genbank manually, you can go on the NCBI website. You can type in the gene of interest. In our case, it's COVID-19. Go. And since it's a popular sequence, it pops up here. The reference genome pops up here. And it has this diagram basically showing in black the length of your sequence, and in green, which areas of your sequence codes for specific things. So for example, the bases from here to here of the sequence codes for the ORF1AB polyprotein. From here to here codes for the ORF1A. And here to here codes for the surface glycoprotein, so on and so forth. So open a new tab. And here we go. Here's the Genbank entry for COVID-19. So the whole lot of this script is attached to it. All right, so the locus is basically, you can define it as basically the name of this gene. And in this case, it's the accession number or ID number, unique ID number of your gene. It is 29,903 base pairs long, and it contains viral RNA, which is expected because it's a virus. And it has been last updated 18th July 2020. Here is a description of what it is. Accession number and your version is just, it has been updated twice in the past. DB link, this is if you want to, it's a separate entry if you want to automatically retrieve it from the net. And so it's a keyword. If you're trying to look for reference sequences, you can type in refseq on your search engine. Source, this describes your organism. And then you have the different times that this specific gene has been referenced in literature. So some of you might notice that some of these references are pretty old, right? It comes from 2005, for example, like way before this gene was even discovered. And that's because this reference here, this reference only references bases 13,476 to 13,503. So what's likely happened is that part of the COVID-19 gene has been found in some other viral genome, and that has been studied already. And that's what this reference means. So you have the references, authors, title, and so on. So here we have a comment from the NCBI staff themselves about the gene. And then we have the different annotations of features of the gene itself. So let's have a look. So the first one here from, here. So the 266 base to the 21,555th base codes for the ORF1AB gene with this locus tag and this database reference number. And yeah, so it has identifiers to do with the protein, and this is the translated protein. So this is in terms of your amino acids, the sequence of amino acids for that protein. And then, yep, so here it describes which parts of that protein string does stuff. So these are annotations of your protein string. And you go down here, ORF1AB again. And here you have your surface glycoprotein from base 21,563 to 25,284. Annotations for that, and so on. And then at the bottom here, we have the actual DNA genome DNA genome for COVID-19, even though it's a viral RNA, but they translate it back into DNA. And yep. So that's what a Genbank format looks like. So if you want to have a go at looking up the databases of the nucleotide databases yourself, then you can have a look at the big three of nucleotide databases, which is Genbank or NCBI, what we just looked at, the DNA Data Bank of Japan, DDBJ, and the European Molecular Biology Laboratory, or EMBL. Right, with that out of the way, what will we be doing for the rest of the workshop? So there's a Jupyter worksheet for you guys to go through in our GitHub, and that consists of 5 parts. So the first part is just importing the data that we're going to use for the rest of the workshop. We're going to count some nucleotides, work with multiple gene sequences, have a go at writing up some code for transcribing and translating DNA sequences, and then having a look at motifs and k-means. So you can go ahead and go onto our GitHub. Maybe someone can post a link on to the chat as well, so people can just click it. And once you're on the GitHub, just go to worksheets and workshop 1, and just download all the files in there. And we'll get started on worksheet 1. If you don't have Anaconda installed, there's a setup guide here for you guys to install it. Yep. So there's steps for you to install Anaconda. And if you have, you can have a go at our worksheets. And yeah, so have a go. If you have any problems or anything, then let us know, and we'll be coming around to help you guys.
Hey, Vincent, I'm thinking we can break people out into breakout rooms potentially.
Yeah. I think, you want to do that?
Yeah. So I think Jason's doing it.
Okay.